Well, today we're going over pheromones, how we're using those in IPM. There's a couple different tactics, but before we get going, a couple of announcements. Again, lab will be meeting in Ag 119 this week. We have Dave Marler as our guest speaker, and we have exam 2 coming up 1 week from today. So earlier I was saying that would cover cultural control through resistance. I decided to cut resistance from it. We'll have that on the next exam. So it'll cover cultural control up until today's lecture, which is on pheromones. So there is a study guide posted to Blackboard. I believe all of the lectures are also posted to Blackboard except this one. So you should have all the materials there. Again, if you get on and you can't find something, let me know, and I will post it as soon as I can. Beyond that, some people came up to me and were asking if I had regraded their labs. I found a couple of them that had been misplaced, so they have now been regraded. All of the available tests are up here. You can just come up and grab it after class, or all of the available lab write-ups. All right. As far as the quiz is concerned, apparently the first question was a little too easy, but I kind of thought it would be. Dispensing large amounts of synthetic pheromone over a crop to mask the pest sex pheromone is an example of mating disruption. Actually, it wasn't 100%. It was listed as 97% correct, but there were no other ones. So I'm guessing someone forgot to click on the first question when they submitted. So they might have missed out on that one. For the second one, this one's a little more challenging. It's the differentiation between pheromones and allelochemicals. So the idea is that pheromones are used to communicate between members of the same species. So that would be intra-specific, whereas allelochemicals are used to communicate with members of different species. So that would be inter-specific. Sort of like international relations is relations between two different countries. Inter-specific relations are relations between two different species.
I think interstates too.
Oh, or interstates, yeah, right? Interstates travel between states. Interspecific is between species. So what we're doing today is we're talking about pheromones, we're talking about allelochemicals, and the reason we're getting at this is that when you are very small, like most pests are, whether you're a weed, an insect, or a pathogen, you interact with the world in a fundamentally different way than we as human beings do. We as humans are very reliant on sound and visual cues to navigate the environment, but when you're something like an aphid, a plant, you're oftentimes interacting with the world on a chemical level. Essentially, you are actively detecting chemicals and using that information to inform your decision-making, your behavioral processes. And what IPM specialists have figured out is that by understanding these chemical interactions and knowing what these chemicals are, we can fundamentally alter the behavior of pests in order to get more control of them. So as far as chemical control is concerned, we're mostly concerned, with pheromone control, we're oftentimes talking about insects, because insects are the most well-studied of these organisms. They're the ones in which we have good chemical-based behavioral control. It's less known in weeds and in pathogens, though I wouldn't be surprised if we head in that direction as we understand these systems a little bit better. So, in general, these chemicals that organisms use to communicate with one another, we call them semiochemicals, or just chemical substances or mixtures that carry a message between organisms. And so whenever a message is being sent, we have to basically designate between emitters, or the organisms that release that message, release that semiochemical, and receivers, the organisms that basically pick up on that particular chemical and then have some sort of behavioral response to it. So they can detect it, and it has some sort of biological relevance to them. And so this will come up a fair bit, the emitter versus receiver, as we start breaking down these different classes of chemicals. And within semiochemicals, we have the two major classes that we talked about just a second ago with the quiz. We have allelochemicals, which are chemical signals that promote communication between different species. So again, interspecific, like interstate. And these can be intentional. So in this example, say you're a tomato plant. you are interested in attracting pollinators to your flowers. So you may release allelochemicals that are attractive to insects that will pollinate you. So that's an intentional interaction with an insect. On the flip side, you may be releasing chemicals that a moth species is using to figure out where you are so it can lay its eggs on you and let the larva feed, which would be an unintentional communication, but it still is an allelochemical, right? It's still a communication between species, whether they meant to do it or not. On the flip side, we have pheromones, which are chemical signals that are used to communicate between members of the same species. And this is what we've been talking about for the most part up until now. Things like moth pheromones used in the traps or used in mating disruption that Forrest talked about. Again, this is intraspecific, so within the same species, and as such, it's almost entirely intentional. You don't have unintentional communication between members of the same species using chemicals. So what we're going to do is we're going to talk about these two different classes, allelochemicals versus pheromones, separately, and then we'll talk about the actual applications of these chemicals in IPM. So allelochemicals are broken up into different categories based on the impact the chemical has on the emitter and the impact it has on the receiver. So essentially it's a question of who does this benefit, who does this harm? Is there one party that benefits more than the other, or do both parties benefit? It's all just questions of the interaction that they stimulate. So the first of these categories are what we call allomones. Allomones are any chemical that's released where the emitter benefits, but the receiver is harmed somehow in the process of detecting this chemical. So essentially, good for the organism that releases it, bad for the one that detects it. An example of this would be maybe with plant allelochemicals, where a plant releases chemicals through its roots that will disrupt the growth of its neighbors. Or on the flip side, insects sometimes will release very aggressive allomones while they are feeding, and this can cause sort of, essentially they can shut down feeding of insects in the neighboring areas, sort of turn them away. A good example of an allomone would be something like the bolus spider. This is a spider that doesn't really catch prey in the conventional sense with a web. What it will do is it'll hang out on a little web it makes, and it's a little hard to see, but what's going on here is it's got one leg extended, and from that it's got a little thread with a bead of adhesive on the end of it, which they call a bolus. Bolus, just for side information, in South America, many of the cowboys essentially don't use the lasso like we do in the United States, but they have something called a bolus, which is sort of a three-ended rope with weighted balls at the bottom. You throw it at the cattle and it will tangle up their legs and cause them to fall over. So this guy's got sort of a little glue bolus. So what the spider does is it hangs on its web and it releases a pheromone that mimics the moth sex pheromone. So the male moths fly up to the spider thinking there's a female moth somewhere, and it snags it with the glue on the end of the web and pulls it in and feeds on it. So it's an allomone because the bolus spider, or the emitter, benefits. It gets to eat. But the receiver, the male moth, gets fed upon, which is generally bad for moths, and so they don't really benefit. They actually are harmed. Kairomones are another class of allelochemicals. This is where the communication benefits the receiver, but not the emitter. So typically, this would be the case of a chemical that tells the pest that there is some sort of food available. When we were talking with Forrest, he talked about how the emitters tend to shut down all of our pheromone traps, right? Because we're flooding the whole orchard with all this sex pheromone, they can't really catch any male moths anymore, and so it's really hard to calculate degree days for some of these species. The way they got around that is what he called a Kairomone bucket, or a bait bucket. And so that's essentially a bucket where you dump a bunch of chemicals in it that will mimic the smell of the host that the organism likes to lay its eggs on. So in the case of like a peach twig borer, you would create a bucket that smells like a rotting peach, and you'll put some sort of stick in it, so the moths will fly up to the bucket, think that there's a peach there, they'll lay their eggs on the stick, and then you just monitor the stick for whenever the eggs show up, and you can use those eggs as your biofix. That way, you're not having any trouble with the mating pheromone because you're swamping out the sex pheromone, but you're not swamping out the kairomone. So kairomones typically are not adaptive to the emitter because the emitter is actually harmed. So typically it's some sort of chemical that the emitter uses for some other purpose, and the receiver, or the pest in this case, has somehow just learned to associate it with the emitter. So if you're an insect pest like the moth, the peach twig borer, they didn't create the smell of a rotting peach to benefit the pest, but the pest figured out that that's what peaches smell like, and that's where I want to lay my eggs. And so in this case, oftentimes the pheromones that species release, the ones they use to communicate with members of their same species, can be co-opted by a predator or a parasitoid and used as a kairomone. So sometimes chemicals can serve multiple purposes depending on who is receiving it and what the ultimate outcome is. So an example of a kairomone would be sort of this whole western pine beetle situation that we're dealing with right now. That western pine beetles are currently, that did turn out a little bit, they're currently wiping out a lot of our pine forests up in the Sierra Nevada, and they are a natural part of this ecosystem. They hang out in the forests, they tend to feed on dying trees, and to sort of kill them off, and they're just a good way to remove older trees from the forest and open up gaps for younger trees to move in. And the way they do that is typically a beetle will land on a tree, start feeding on it, and the tree will go through a response, a defense response, where essentially they'll produce a whole bunch of sap, and they will push the beetle out of the hole that they drilled into it using this big sap plug. So here you see a beetle drilled a hole, the tree pushed out all of this pitch, and you can see the beetle is stuck in the pitch being pushed out. So a single beetle can't really feed that successfully on a tree. But in this pitch, there's a chemical called myrcene. And myrcene has been co-opted by the beetle as basically a sign that there's a sick tree somewhere with beetles feeding on it. We should all aggregate there and feed on that tree. And so all of a sudden you go from having one beetle on a tree to having several hundred of them all drilling holes into the tree. And if the tree is sick, the tree can't produce enough pitch to push them all out. And so the beetles will eventually kill the tree, reproduce, and go out to find new trees. Now the reason the beetles are having such a huge effect right now is just that we're in the middle of a drought. So there's lots of sick trees that can't produce enough pitch to push them all out. Well, we were in the middle of a drought. Either way, there's still a lot of dead trees out there. And then the last of the allelochemicals are the cinnamones. Cinnamones are great. They benefit everybody. The emitter gets a benefit. The receiver gets a benefit. And a good example of this would be something like a really cool system that's been figured out by a team in Germany. They've been researching this in great levels of detail. It's called wild tobacco, and they basically figured out that when tomato hornworm or tobacco hornworm feeds on this wild tobacco species, that the plant will release a whole bunch of what they call green leaf volatiles. So these are just sort of alarm chemicals that tell other plants in the area that there is something feeding on it and that they need to watch out. So it's a pheromone for other tobacco plants. However, these green leaf volatiles also are attractive to geochorus bugs, or big-eyed bugs, which are a natural predator of these hornworms. So functionally, it brings in these geochorus bugs, they feed on the tobacco hornworms, and the plant gets some relief from all the feeding it's receiving. So this is a cinnamon because the plant gets benefited that it is protected from the pest, the hornworm. But then on the flip side, the geochorus also gets a benefit because it's attracted to a food source. So this is a chemical is a win-win for both organisms. And there's a handful of situations like this in the plant world that oftentimes plants will release volatiles in response to insect feeding that are generally attractive to predators and parasitoids of those pests. So that's it. We've got allomones, kairomones, and cinnamones. that all just differ in who benefits and who suffers for the release of the chemical. And then on the flip side, we have pheromones, which again are used to communicate between members of the same species. So generally, all pheromones are designed to benefit everybody who receives them and to benefit the emitter, because there's no sense in releasing a chemical that's going to harm you from members of your same species. Generally, pheromones are just classified by the response that they elicit whenever they are received. So you have things like alarm pheromones, which are released whenever there's danger. It warns the rest of the population. So an example of this would be lima bean plants. When lima beans are being fed upon by spider mites or many other sort of piercing-sucking insect pests, they release a whole bunch of those green leaf volatiles, and those tell all the surrounding plants that there's some sort of pest in there feeding on them, and they will all upregulate their defensive pathways to prepare for the pest to move over to them. So just a nice little warning to the others. Similarly, in alarm aphids have alarm pheromones. So for certain aphids, if you go up and squish one of them on the plant, all the other aphids will drop off because they think there's some sort of predator there that's going to feed on them. Aggregation sex pheromones, like we've talked about before, they are attractive to, basically they attract members of the same species, either to mate or it tells them that there's some sort of prey item there that they can all go and feed on. Excuse me. So that would be also an example of the western pine beetle, that not only is it attracted by the myrcene, but they also release aggregation pheromones to attract more of the beetles to that same feeding site. And then the food trail, right? So ant species especially, when they're feeding, they'll just sort of drag their hind end on the ground behind them to leave a little trail of chemicals that will tell other ants, this is where I went. There's a famous entomologist / evolutionary biologist, E.O. Wilson, who figured out how these pheromones worked. And he used to have a little parlor trick he'd do where he'd take an ant and he'd pull out the little gland that had it. He'd sign his name in the pheromone, and the ants would all swarm all over it and form a little moving ant line in his name, which really impresses people at entomology conventions, I suppose. But this leads to a sort of an interesting side behavior, which is that sometimes what ants will do is they'll accidentally cross their own pheromone line, and then the trail out of that pheromone loop gets disrupted. Maybe someone kicks it over, or maybe there's a little rainstorm. And so they'll end up in this big sort of loop where they're just following each other around in this big circle. And in some cases, the ants can't ever get out of it, and they can't go back to find their nest. And so they'll just walk in this circle until they all die of starvation.
Literally a death spiral.
Sorry?
Literally a death spiral.
It is literally a death spiral. So you'll see this every once in a while. All right, so how do we apply these chemicals in IPM? What is the actual application? Well, fortunately, pheromones are really well studied. There's been a lot of work trying to identify what pheromones are associated with different pest species. And so like Forrest mentioned when he was at lab, we have essentially online databases with thousands of different pheromones for different species. And ideally, by synthesizing these pheromones in large quantities, we could basically throw insect behavior into some sort of overdrive. And ideally, in a way, that will cause them to die out, or at least to avoid the crop that we are trying to protect. However, there are some caveats to this, that it's not always super easy to synthesize these chemicals in large amounts in a way that's attractive to insects or causes the response we want, because insects are highly, they're very sensitive to using the exact right chemical. And even when you're using the right chemical, you have to make sure it's the right stereochemistry or the right enantiomer that you are using. So just as a reminder, you all have taken biochemistry or should be in the process of taking biochemistry, right? All right, so stereochemistry is basically the idea that when molecules are put together, right, due to their electron structure, Oftentimes chemicals with the same exact formulation can have different modules of the molecule pointing off in different directions from one another due to things like double bonds in the carbons. And so oftentimes what you'll have is two chemicals that are identical to one another chemically from a formula perspective, but they don't have all of their different groups in the exact same formation, right? So they will look like mirror images to one another. Sometimes we call this handedness, the idea that your hands are exactly identical to one another. You've got your 4 fingers and your thumb, but if you try to lay your hands directly on top of one another, it doesn't work. You've got your thumbs on the opposite sides of your hands. The hands are actually mirror images of one another. So they fold over, bottom to bottom, but you can't put them on top to top. And there's lots of chemicals like that. If we tried to overlap these two chemicals with one another, the COOH would end up over here, this guy would end up over here. Essentially, they just don't match. And so ultimately, for a chemical to work in a pheromone setting, you need to make sure that all of your chemicals have the right handedness. They need to have matching stereochemistry. Yeah, I threw that in there. That was a different example. So just to give an example of how much stereochemistry can matter, there's the chemical Kairomone. So Kairomone is this chemical right here. It's a little, sort of benzyl ring with a double bond and this little extra group hanging off the side. And depending on whether this hydrogen is attached in the R configuration or the S configuration, whether it's pointing down or up, you get two very different chemical properties. R-Kairomone produces the smell for peppermint, whereas S-Kairomone produces the smell for caraway seeds. So it's kind of the difference in smell between a peppermint patty and a slice of rye bread, that it's chemically identical except for the direction some of the groups point, and it produces a hugely different response. Now this matters in insects because insects are very good at differentiating between these different enantiomers or these different stereochemistries as well. So, for example, they figured out the gypsy moth pheromone, but when they put it out in trials, it had virtually no attractiveness because they were using the S enantiomer instead of the R one, which is the one that is attractive. And in some cases, insects can be really frustrating because they actually need a specific blend of these two enantiomers in order to recognize it as their sex pheromone or their aggregation pheromone. This guy, the western hemlock stainer, again, another forest pest, it needs to have a 65-35 blend of the R and the S enantiomers, or else it won't recognize the host. And so oftentimes it can take a lot of experimentation of trying different blends at different concentrations with different enantiomers before you find the exact right one that matches the species. And this is probably why, like Forrest said, I think they worked for 3 years trying to figure out the red scale pheromones. And it's probably because they had it on the database, they knew what the chemicals were, but they had to try a ton of different blends before they found the one that actually matched the species. So within IPM, we have 3 major uses for these pheromones. We've got detection and monitoring, like we've been using in the lab. We've got mass trapping, and then we've got mating disruption. And generally, all of these are reliant on sex pheromones. We're typically not using trails or alarms or any of that other stuff. We're really relying on basically using sex against the insects. So just a quick review, how sex pheromones work. In general, with insects, sex pheromones are produced by the female in order to attract a male. It generally does not work the other way around. Males typically don't produce anything. So males will ultimately find females using a behavior called trail following, in which they will essentially detect the pheromone and fly up the plume to find the female. So females produce the pheromone, typically sitting in some location where the wind can carry it, and they produce really minute amounts of this. Males have been known to detect pheromones at as little as 1 × 10 to the -9 grams per centimeter. So that's, I can't even put that in context how very little that is as far as concentration. it's more or less along the lines of about 1 parts per billion in the air. And males can detect at much lower concentrations than the females even release. So once they release these pheromones, the wind distributes them out into the plume, and then the males will detect it and fly upward towards the source. And males, basically what they will do is they'll use a technique called crosswind casting, where basically whenever they detect a pheromone in the air, they will turn and fly towards it whenever they detect it. So they'll turn into the most concentrated part of the plume, and they'll just keep flying straight until they hit a point where the plume starts to thin out and they can't detect it anymore. Then they'll just cut back the other direction, essentially zigzagging up and down the plume until they get to a point where they actually find the female. So the first way that people figured out to use pheromones was just through monitoring, like we're doing right now, where essentially you have a combination of a sex pheromone with some sort of trap that catches the insect. The insect flies up the plume, thinking there's a female, gets caught, and you can use that as a way to establish whether a pest is present, what life stage they're in order to calculate degree days. There's a handful of applications that are useful. And generally you would use sex pheromones, but like we mentioned before, you can use Kairomones. You can trick the pest into thinking there's some sort of food source available, and they'll fly towards that. So you can use this for the biofixes. Alternatively, some people have traps set up like this where they measure the number of individuals in the trap, and they'll use that as a trigger for a pesticide application, such as codling moths. And then this is widely used by the US government to monitor for invasive pests. So if you go down to like LA County, all around the ports, they've got these little bucket traps that are used for medflies, and they'll just set them up all year round and periodically go out and check them to see if there are medflies. And if they ever detect medflies, they'll just dump the whole area with sterile medflies to swamp out any of the invasives and cause them to die out. Here in the Central Valley, we have a concerted monitoring program that uses these for pink bollworm. So pink bollworm is a major cotton pest. It's widespread throughout Mexico, the Southern California deserts, and Arizona, but it never really established real well here in the Central Valley. And so the Central Valley growers basically said, well, let's keep it that way. So what they did was they established a grid where they set out a pheromone trap about 1 every 600 acres in the Central Valley. These are periodically checked over the summers, and if there's ever a male found in one of these traps, they just inundate the area with sterile male pink bollworms in order to sort of flood them all out. And so, yeah, they'll fly a plane over, and here you see a little plume. That didn't turn up real well, but you can see the sort of dark area. That's a plume of male moths being dropped out into an area where they found some pink bollworms. And what's kind of clever is that when they raise the pink bollworms, they'll actually feed them on a diet with some pink dye in it. So the male moths that are produced are extra dark pink. And that way, if you catch a sterile male in the trap, you can tell the difference between sterile males and non-sterile males, and you can, still keep monitoring an area effectively. And then there's mass trapping. So mass trapping is essentially just setting enough pheromone traps out there that you actually severely dent the population, right? So if you're already attracting males to a trap, why not just set so many traps that you attract all the males, they all get stuck and they all die? Well, the main reason you don't do that is that there are oftentimes so many insects in the system that you can't possibly catch them all. You can only catch a very small fraction of them. But there are some systems where this has been used effectively, that you can design these sort of behavioral controls that do catch the majority of the pest. And typically you can do this either through just having a massive amount of sex pheromone in the system or using some sort of aggregation pheromone in order to catch both the males and the females. So an example of this being used successfully was with the American palm weevil. So this is a big pest in South and Central America. They've recently shown up in Southern California as well, where they cause trouble to palm trees that are like in people's yards and the like. But in South America, they're a huge problem because there are these large palm plantations where they're raising massive amounts of palm oil. And so essentially in the 1990s, they had a problem where these weevils would move into these palm plantations, they would feed on the trees, and they would transmit this disease called the red ring disease, which can kill the tree in a matter of 1 or 2 growing seasons. And the infestations were bad enough that most growers were tearing out 15% of their trees every single year and replacing them due to this. So people were just spending a ton of money. And ultimately what they came up with was a technique in which they put what they call an attracticide. So you create a blend of, I believe this was it was the male pheromone. So they put a sex pheromone in there, mixed in with a whole bunch of sugar cane, so it also is attractive to females as a food source, and then they dumped a whole load of pesticide in there as well. So essentially you're attracting males and females to this bucket, they go in, they feed on the sugar water solution, and they die due to the ingestion of the poison. And you set out about 1 of these for every 16 acres, and within the first year, they trapped this ridiculous number of males, about 200,000 of them over 40,000 acres. And ultimately, what that led to was a reduction in tree removal by about 99%. So they went from 10,000 trees being yanked out every year on some of these plantations down to about 50. And this is just a chart of that. It's the number of red ring death palms, red ring disease palms that were yanked out. You can see that in the early 90s, they kind of peaked out at this 10,000 point. Then once they started doing this attracticide program, they knocked it down to about 50 by the early 2000s. I was just talking to a guy from Trinidad, and he was saying they tried something like this on the islands, but they made a mistake that they didn't use nearly enough buckets. They figured they could put out just enough to sort of stop them from moving into the palms from the forest. So they set up a couple buckets near the edge of the forest. But it actually had the opposite effect of what they wanted, that it acted as an attractant, pulling in thousands of beetles from the forest, which then just moved into their palm plantations. So if you're going to do something like this, you have to be in it 100% all the way. Yeah. And in the US, they used a similar project to this to eliminate boll weevil from the southeastern United States. It was fairly successful. So generally, for these mass trapping programs to work, you need to have a couple of things that align. On one hand, you need to have species that are present in relatively low concentrations. If you're trying to use something like this to control aphids, where you have potentially thousands of them on every single plant, you're never going to set out enough traps to catch them in a cost-effective way. You would just need a ridiculous number of traps. So you need something like the beetle that's relatively large. You don't have a whole ton of them in any one location. You need adults that are relatively long-lived so that you have a better chance of capturing the adults. Certain insects like scales are really good at putting out a ton of males all at once. They mate very quickly and then they die within 1 or 2 days. So if you have something like that, it's going to be really hard to attract those scales to a trap in such a short amount of time. And you also need adults that are very strong flyers and have a pheromone that they orient towards. Again, for the same reason. You need them to be able to travel all the way to the bucket to get caught. And if they aren't strong flyers, they won't make it. Similarly, if there's no pheromone, then what are you even doing? It doesn't make sense. And then finally, we have mating disruption, which is becoming increasingly popular in the valley, like Forrest was talking about. Of course, Forrest works for a company that sells mating disruption devices, so it would make sense that he would say that. But if you've driven around the valley, you've noticed these tags. I know a number of you have worked for companies that use the puffers, so this is becoming increasingly popular. So as far as mating disruption is concerned, there's really 2 major tactics that are used. There's the idea, on one hand, of what we call false trail following. So the idea of false trail following is what we've got with these little strings or tags that people put out. Essentially, you put out many little point sources of pheromone, and these act like false females. And so the males will detect their pheromone plume, fly up, and try and mate with the tag. And that will essentially just sort of dilute mating, or at least slow it down so that mating doesn't happen immediately. So essentially, you're just wasting the male's time and you're wasting their energy. And sometimes what people will do is they'll actually build a tag that also has a little bit of pesticide in it. So the male comes along, tries to mate with it, and gets a little bit of a dose that could kill it off. Though that's not super common here in the valley. So essentially, right, you got a male following a trail, but if you've got multiple females, it's really hard for the male to follow it correctly, and the location it may choose is likely to be a false source, not a female. On the flip side, you have mating confusion. And mating confusion is a little bit closer to what we see with the puffers. Instead of setting out a whole bunch of false females, what you do is you just release very large doses of pheromone into the field in sort of a regular interval. So that if you have an almond orchard where normally you'd have multiple plumes floating around with the pheromone, instead now you've got an almond orchard where all the air is effectively one huge pheromone plume. It's kind of like if you go into a barn and at first you notice that the barn smells really bad, right? Maybe you go into a cow shed and you notice, oh, there's all this manure smell, it's really unpleasant. But the longer you stand in the manure shed, the less and less and less you notice that particular smell because you're getting used to it. And sensory organs are designed to detect differences in the environment, not to always detect the same thing being exposed to over and over and over again. So you get habituated. And that's the idea here. You're putting so much pheromone out there that the insects are getting habituated to the amount of pheromone in the environment. So they can't tell if they're flying through a plume or not because everything smells exactly the same to them through their antennal receptors. They're basically just used to a certain amount of pheromone being around. And so ultimately, you get the same result. The males are flying around, they can't detect the plumes, and so they can't find the females. Right? So essentially, same idea. You've got so much, this isn't a really great example, but in this case, they've got so many plumes that the male just can't even find the female at all. So, when this technology was first being introduced, there was a lot of people who were wondering, how does it actually work? Because they knew that they had lower populations when it was happening, but a lot of people were seeing that they were getting the exact same amount of egg laying going on as they were without the technology. So they were seeing the same number of egg packets, but fewer moths. And so essentially what they did was a study in the 90s where they looked at these oriental fruit moths, and they demonstrated that by following these moths around, measuring their mating rates, that this use of mating disruption didn't actually stop them from mating very much. You only saw a 15 or 20% reduction in the amount of mating. But what it did do was it delayed the amount of time it took for a male to find that female. And that did a huge amount of difference. And this is because female moths, when they emerge from their pupa, they come out with a set number of eggs that they can fertilize, and they don't produce any more. And the longer the female moths are alive, they need energy, and they typically don't go out there hunting for food sources. So what they'll do is they'll start reabsorbing their eggs and using those eggs as energy. So if you can delay the amount of time between the female moth emerging and when she gets mated, she may have absorbed half of her eggs. And all of a sudden, her clutch size is just 1/2 of what it would have been without that mating disruption. And so you're not even so much knocking down a huge amount of mating, although 15 to 20% reduction isn't shabby. But also those that do mate are producing significantly fewer offspring than they would be without this technology. So it's kind of a double whammy from the moth's perspective. All right. So as far as pheromones are concerned, there's a lot of upside. They are being used very effectively in the valley, but there are some limitations. One challenge is that if you are planning on using pheromones, you need to know that you're going to use them before you know there's a pest problem, right? So pheromones only work if you apply them before the pest emerges, before they go out to mate, and so you can't actually really monitor for the pest and make a decision to use pheromones then. You have to just sort of assume the pest is going to show up and use the pheromones preemptively. So the challenge with this is just that it puts the cost up front on the grower. that basically the grower needs to say, I think we're going to have oriental fruit moths this year, so we should just use these pheromones. And if you have fruit moths, then great, you came out ahead of the game. But if fruit moths never show up, then you just spent money on a control tactic that you didn't need to use. Generally, you need to apply these techniques area-wide, especially if you're doing something like an attracticide bucket. It's not just good enough that you're disrupting mating in your orchard. If your neighbor who's upwind of you isn't doing it, then the mated moths might just fly into your orchard and lay their eggs there. And so generally, these things work better if everybody's on the same page and you're using it, your neighbors are using it as well. Again, pheromone blends are extremely species specific. And so, while some species we may have really good technologies like the navel orange worms or these California red scales, it may take a really long time to figure out the exact blend. I was talking with Kent Dane, a researcher down in Parlier, and he and his team have been working for a while to come up with a pheromone blend that works on lygus bug for cotton control. And so they had a really nice paper. They've been working with this guy who knows exactly what pheromones that these lygus bugs use to attract males. And so they created all of these different blends using these chemicals. They came up with one that worked really well. They released it in the field, and it turns out that it's super attractive to the least common type of lygus bug, not the Lygus asperis. It attracts this one species that shows up like 1 in every 1,000 lygus bugs. And so they'll set the traps out. It'll attract this really rare species until their sticky cards are completely clogged up with them, and it has no discernible impact on the Lygus asperitas, the one that causes the most damage. And so they're kind of back to the drawing board now, that they developed a really good tool for something that's not a huge pest, which is unfortunate. And then, I should probably take this one out, because back in the day, this was relatively expensive, but like Forrest was saying, these technologies are becoming increasingly inexpensive. And so a lot of growers are getting on board in comparison to pesticides.
You say about $20 an acre, right?
Yeah, so very reasonable. So with that, that's an old slide. We're not going to do chemical control next. We're going to move into mating disruption. I'm sorry, not mating disruption. Pesticide resistance and resistance management. Again, resistance management will be on the final exam, not on this upcoming one. So if you study from here all the way back to cultural control, you'll be good for next Thursday. So with